So the next bunch of slides are the organelles that we talked about in the lab. Were there any questions about the organelles? Okay. So, was it the last one? Yeah, the last organelle that we talked about were the centrioles or the centrosomes. And when we talked about these, we said that they were the attachment for microtubules. And we learned that they were kind of skeletal structure, but we didn't talk about them all that much. So microtubules are part of what we call the cytoskeleton. Again, cyto is cell, so this is going to be the skeleton that gives our cell structure. This is going to be made up completely of proteins strung end to end to end to end. This is going to give our cell a shape. It's going to organize and hold organelles and things in place. They're going to act as highways to move vesicles and proteins different parts of the cell. They're going to also be important in cell movement. We don't like to think of our cells usually as moving, but there are cells in our body who move a lot. Fibroblasts are a type of cell that actually crawl through our skin looking for wounds. When they find that wound, then they'll set up shop, and then they'll stay there and become a skin cell, basically. But they need to move, and that's going to work by moving the skeleton. You push a skeleton forward, it's going to create a blob going forwards, and then you dissolve the skeleton in the back. While that's happening, the cell is actually going to crawl forward. There are 3 different types of filaments involved in the cytoskeleton. There are going to be microfilaments, intermediate fibers, and microtubules. We'll talk about each one independently. So this is just a generic picture of our cell containing the cytoskeleton. And so we have our nucleus, we have our cytoplasm, we have the cell has microvilli, the little fingers up here, and then there's going to be the cytoskeleton here. And so microfilaments are going to be the first one we'll talk about. That's on, I believe, the next slide here. They're going to be more or less up near the cell surface. They're going to make what's called this terminal web. They're going to form the outside skeleton of our cell. And then there are intermediate filaments, which are very, very small, but they create this gigantic mesh through the entire system. And then there are these very large microtubules. Microtubules do have a support system or support function to them, but the microtubules have more directly functional roles. They're going to act as the highway through the cell, and they're going to be very, very, very important when we talk about cell division. So the first one was those microfilaments. Those are the ones at the outside edge of our cell. They're just going to make this little mesh wire going around the outside of our cell. Each one is only about 6 nanometers thick. So a nanometer is a billionth of a meter. So take a billion of them laid next to each other in order to make a meter. These are made of actin, but that's an important protein. Actin is an important protein in our cytoskeleton, and it makes that terminal web. Slightly larger than the microfilaments are the intermediate filaments. These are 8 to 10 nanometers. These are primarily within our skin cells, and these are made of keratin. Microfilaments are made of actin, intermediate fibers made of keratin. These are going to give the cell a shape, and they're going to allow it to bounce back when you compress them. And so you always see commercials on TV for skin creams talking about keratin and making your cells, your skin supple and flexible, right? The idea, at least in principle, is that keratin makes this intermediate filament which makes your cells kind of springy. And so that's how it's going to resist stress. Then the big ones are microtubules. These are 25 nanometers, and so much larger than the other two. These are made of a protein called tubulin. So we have actin, keratin, and tubulin. These are going to grow out from those centrosomes. The centrosome was that organelle made of two centrioles. Microtubules are going to come out of that centrosome, and these are going to come and go. Whereas microfilaments and intermediate filaments get made, and then they're more or less permanent until they just fall apart from old age. These are going to constantly be getting made and broken down. Like I said, they do help maintain the cell shape, but the primary purpose is to move things through the cell, so to act as railroad tracks for these motor proteins. The motor protein is going to walk down the microtubule, carrying something with it, and then it also makes what we'll see these axonines. These are very important in cell division. If you take a cell and you stain cytoskeleton, this is essentially what you're going to see. It's just absolute mesh of proteins. All these little strands are proteins. So there's one cell, there's another one, and another one, and another one, and another one. And it's just full of these proteins. It gives it shape. So this is what a microtubule really looks like. It's not just one strand. It's actually a bunch of strands arranged in a tube. Your microtubule is a tube. It is hollow. You don't need to know the name for each individual stick, but those are called protofilaments. These dynein arms, again, you're not responsible for that. That's that motor protein that's going to walk along the microtubule carrying something with it. I think this is a good place for a break. So we'll start at 25 after on that clock, so almost 15 minutes. Hey, keep it going. It's not like you can get on my good side, right or wrong. The more likely to imagine you die. An organism dies. It's no longer eventually the cells in its body are individual little divisions. So if an organism dies, the cells are perfectly fine. So they're going to use up all our nutrients in themselves, and then they're going to use all the nutrients in the one vascule. And then they're so all of a sudden so the potassium pump can't work. So the potassium pump stops working, and then all of a sudden eventually so the glucose pumps out. So it can't bring any more glucose. We're anaerobic still uses glucose. It's not sufficient for it. So for anaerobic you get 2 ATP. Aerobic you get 36. So no situation per cell. Right, but when you run out of glucose, next thing we're going to talk about is our DNA. So we're going to really change gears here. We're going to talk about what DNA is, and then we're going to talk about genes in cell division. So you all know that DNA is a double helix chain. So this is really what it looks like in just drawing form and then kind of more of a molecular form. It looks like a spiral staircase, right? With the 2 guardrails and then the steps going up it. The 2, what here they call side pieces, are an alternating series of phosphate and sugar. So remember, our nucleotides that make up DNA are made up of a phosphate, a sugar, and a nitrogenous base. And so the sugar and the phosphate just alternate along the DNA backbone, and then the nitrogenous bases are here. These are the important part. These are the coding part. These are just the structural part over here. So every nucleotide is going to have the same phosphate and sugar. It's the nitrogenous base that's going to be the same. So DNA is going to be organized in a number of different levels. DNA is actually a monstrous molecule. And our cells need to be able to pack a bunch of it into each tiny little cell. And so if you look at double helix, so right here it's saying the diameter of that is about 2 nanometers. And it's going to get wound down, and it's going to get wound around a protein. That protein in DNA with DNA wound around it is going to be called a nucleosome. DNA wrapped around this protein is called a nucleosome. And then the nucleosomes kind of pack amongst themselves. and then the DNA kind of folds on itself, and then it folds on itself in kind of a spiral pattern again, and then you end up in a dividing cell with a neat looking chromosome that looks like this. And so a chromosome is going to be that molecule as a whole. The term chromatin is going to be used more as a generic word to describe the material that goes into making a chromosome. And so the chromatin is this combination of the DNA and the protein in the nucleosomes. So the chromatin is the DNA material complexed with the protein. Our bodies have 46 chromosomes in each cell. There are 23 pairs, so there are 2 copies, one from each parent for each chromosome. Although each chromosome is actually a 6-foot long molecule. So you have 46 6-foot long molecules packed into the nucleus of each tiny little cell. In a non-dividing cell, the chromatin is not compacted like that, where you can really see it. So what you're going to have is a nucleus with a whole bunch of randomly strewn around chromatin that just looks like a nucleus full of DNA. You're not going to see individual strands. If you have an electron microscope, so you can actually look at it at the almost molecular level, you're going to see something like this, where you see a strand and a dot, and a strand and a dot, and a strand and a dot. In order for a cell to divide, it has to copy its DNA. So we have 46 chromosomes. If a cell divides, it needs to keep 46 for itself and then give 46 to the new cell. So we have to copy every chromosome in our cell before it can divide. And there are 4 steps in that process. We're going to have to unwind the double helix. We're going to have to unzip that DNA helix. We're going to have to make new DNA, and then we're going to have to package it all back together. So unwinding is taking it off of those histones. So we have that DNA strand wrapped around a histone. We need to unwrap it off that histone. And then we need to unzip that double helix. We're going to duplicate each strand independently. We're going to take these 2 apart. We're going to duplicate this one, duplicate that one. We're going to end up, now we have 2 copies. This one will go to that cell, this one will go to that cell. And so it has to unzip it. Where it is unzipping, the point where it is actually unzipped, is called the replication fork. So replication is the copying process. The fork is where the DNA is breaking apart. Second part is the replication itself. There's an enzyme called DNA polymerase. It's ASE, meaning it's an enzyme. It's going to make a DNA polymer, so it's DNA polymerase. It's going to move down that strand that is now opened up, and it's going to read each base as it goes and put in the complementary base. And so if we have a strand of DNA, let's say we have A, T, G, and this is going in really far in each direction, and we have our polymerase here. It's going to see an A, and it's going to put in a nucleotide here that is complementary to A. What always pairs with an A? It's a T. And so what we're going to end up with is we're going to have an A and then a T. And then it's going to move on to the T. What's it going to put with a T? A, and then it'll move to a G, C, and then these are all going to be attached. So now we have a double strand. When we broke the DNA apart, there was already a TAC So originally, this strand was attached to that one. We broke it apart, Now we're going to copy this one and copy that one. So remember that you go AT and CG. If it sees an A, it'll put a T. If it sees a T, it'll put an A. C goes with G. And so you'll have two separate polymerases, it says. We got one working on this strand, you have another one working on this strand up here. And so they're going to more or less finish at the same time. And in the end, you're going to have two complete double helices. And so each one of these will have the two strands making it up. We call it semi-conservative because each new strand of DNA contains 1/2 of the old original copy. We had one copy, we split it up. Now, half of this strand is old, half of it is new. Half of that one is old, half of it is new. And so in the end, these will get sent to two new cells, and then when it divides again, half of this will end up in one cell, half will end up in the other cell. And then once it's made, we have to repackage it. It takes about 6 to 8 hours in order to copy all of our chromosomes. While that is happening, we have to make millions and millions and millions of those histones. The proteins that the DNA is going to get wrapped around, they're going to get made, and then once the DNA is finished, it's going to get wrapped or back around those histones, and then we're going to have something called a nucleosome. The nucleosome is a bunch of histones together. This is a look at. This is the original DNA strand. That's the DNA polymerase. So it knows where it's going to start. It comes in. Actually, sorry, that's not the polymerase. That's something called helicase, which you don't need to know. But that's what actually unzips the DNA. Now he's showing you at the molecular level what's going on. It sees an A, so it's going to bring in a T. And this DNA polymerase that's doing that. There's a lot of details here you don't need to know. As it moves, it gets unwound and copied. And then at the end, we have a strand being made here, and a strand being made there. And then once it's finished, we'll have two new strands that can be packaged and put into the other cells. So cells have a specific life cycle that they always have to go through. We call that the cell cycle. There are two main parts to that cell cycle. There is interphase and mitosis, and then each of those pieces has subparts. And so in this cell cycle, it's just going to keep going around in a circle, doing each of these phases as you hit it. So interphase is going to be everything in the green here. That's the parts where the cell is not actively divided. Mitosis, or the mitotic phase, is where the cell is actively divided. Interphase is made up of 3 subphases, G1, S, and G2. S is synthesis phase. DNA gets replicated in S phase. Nothing of great importance happens in G1 or G2. They're called gap phases. The most important things happen in S phase and mitosis. And so G1 is gap 1. It's the gap between mitosis and S phase. G2 is the gap phase between synthesis and mitosis. Mitosis has 5 subparts that we'll talk more in depth about, prophase, metaphase, anaphase, telophase, and then actually cytokinesis is the fifth one. So mitosis is the process of taking one cell, copying those DNA molecules, and making two daughter cells. This has a number of purposes in our body. Number one, it allows us to go from a single cell, when we first start out, to the 50 trillion cells currently in our bodies. We use mitosis to replace cells that die. So our cells are constantly dying, we have to replace them. So for every one that dies, one has to divide to make a new one. And this goes along also with damaged tissue. If I have a wound in my skin, the skin cells around it are going to have to divide to make new cells to fill in that hole. So we are not going to talk about meiosis in this class. Meiosis is part of the reproductive system. The only cell division we'll be talking about is mitosis. But if you know anything about biology, you know there's mitosis and meiosis. So you're going to have to remember that mitosis is the one that we deal with. It's stupid, but the way I remember it is mitosis is used for damaged tissue, right? So if I stub my toe, you're going to have to do mitosis to repair it. That is literally how I remember it, okay? If you just know it, great. But if you can use my stupid little thing to help you, great. So mitosis itself has 4 phases, and then there's cytokinesis, which some people consider part of mitosis. Some people say it's actually kind of its own little thing. So the first part of mitosis is prophase. And before I talk about this, you're going to have to know the order here. The way a lot of people remember this, including me, is PMAT, P-M-A-T. If you can remember PMAT, and you remember what the 4 are, then you know which order they go in. You're going to have to put them in the correct order, and you're going to have to know what happens in each step. So the first one is prophase. In prophase, that loose chromatin in our nucleus is going to condense down into those X-shaped chromosomes that we saw in that picture. In prophase, you are going to be able to look under a microscope and see individual chromosomes. You'll be able to see 46, and each one will be this sort of X shape. That is an entire chromosome made up of 2 individual copies. That is identical to that. Each of these is called a chromatid. The whole thing is a whole. It's called a chromosome. Each identical half is a chromatid. You might see it as a sister chromatid. In prophase, the nuclear envelope is going to break apart. In order for these chromosomes to get pulled into 2 different cells, the nucleus has to fall apart. Otherwise, the DNA can't get pulled apart. While this is happening, the centrioles are going to start putting out long microtubules. They're going to reach out, and they're going to grab this connection point on each chromatid. That connection point is called the kinetochore. So you have chromosomes, chromatids, kinetochores, and then you also have these, remember these are coming off of centrosomes made of centrioles. There's a lot of words that sound similar here. You're going to have to make sure you know which one is which. And so, here's a picture of prophase. So here's our nucleus. It's starting to break down, and we have individual chromosomes that are here. And the little dots, purple dots here, are the kinetochores. Each chromosome is made up of 2 chromatids. We have our centrosomes, each one containing 2 centrioles, and then you have the microtubules coming off These two centrosomes are going to move one to each side. And then once prophase is finished, we're going to move on to metaphase. In metaphase, all of these chromosomes are going to line up right down the middle of the cell. And this line going through the middle of the cell is going to be called the metaphase plate. The chromosomes are going to line up on the metaphase plate. The spindle fibers, which are the microtubules coming off of the centrosome, are going to attach to the kinetochores on the chromosomes. So you can see that each chromosome has a spindle fiber coming from each centrosome. What eventually is going to happen is these are going to pull that apart. The centrosomes are also going to send microtubules the opposite direction to anchor them to the plasma membrane here. And that is called the aster. So the microtubules coming off the centrosome, connecting to the plasma membrane as an anchor, is called the aster. So we've got the chromosomes along the equator of the metaphase plate, spindle fibers. So this whole thing, all those microtubules connected to the chromosomes, is called the mitotic spindle. Each one is a spindle fiber. As a whole is called the mitotic spindle. And then you have some microtubules that go backwards, anchoring to the cell membrane, and that's called an aster. So now we're ready, everything's attached, we're ready to start pulling apart. Next is anaphase. In anaphase, there's an enzyme that's going to break apart these two chromatids. Without that enzyme, these cannot pull strong enough to break them apart. Something's going to come in and cut them, allowing the two to go apart. And so now each chromosome, made up what was each sister chromatid, is now each a chromosome, and it's going to go to each side of the cell. So this is anaphase. In anaphase, the chromosome is broken into halves. Each half is going to one side of the cell. And then you can see that the cell is just barely starting to make this kind of fold here, where you can see where it's going to divide in half. So that is anaphase. And then in the next part, we have telophase. In telophase, the chromosomes have moved to each side. So now we have chromosomes in this cell and chromosomes in that cell. The rough ER is going to kick it into high gear, and it's going to make the nuclear envelope again. The phospholipids that make that membrane for the nuclear envelope come from the rough ER. So we're going to make the nuclear envelope again. The chromosomes are going to fall back apart from nice, visible individual chromosomes to this kind of nebulous little blob in the nucleus. The mitotic spindle is going to fall apart. It's going to just completely disappear. And then the nucleus in each new cell will make the nucleoli, the dark spot inside of the nucleus where the ribosomes are made. So this is telophase. In telophase, nucleus reforms, DNA uncondenses, and the cell is just about to break apart. The actual process of breaking apart is called cytokinesis. So telophase brings it to the point where it's just about to fall apart. Cytokinesis is the actual division of the two. Telophase and cytokinesis are kind of fuzzy, where one ends and the next one starts. But cytokinesis is the actual finishing process of breaking the two cells apart. And the way this works is there's a protein called myosin, which is very important when we talk about muscle. It's going to pull on the actin in the terminal web. That was that mesh just around the cell membrane. It's going to pull on that. You can imagine if you had something that was pulling this towards that, it's going to pull it together until it meets in the middle, and then the membranes are going to fuse and then break apart. cleavage furrow is where it's about to break, where it kind of folds in on itself. It's called the cleavage furrow. And then eventually it breaks into two, and then we have a video showing this whole thing together. So this is a cell in interphase. So the DNA is uncondensed. Now we're in S phase. We're going to replicate the DNA to make an extra copy of each chromosome. We didn't talk about it, but it actually has to replicate the centrosome also. But eventually you need one in each new cell. So now we have two of the centrosomes. Our DNA is condensing. Eventually we're going to end up with individual chromosomes. Our centrosomes are now migrating to each side of the cell. Plasma membrane, or on the nucleus, the nuclear envelope is starting to break down. The mitotic spindle is forming. Now that the envelope is really breaking down, the chromosomes are migrating towards the middle. Mitotic spindle is attaching to the kinetochores. In metaphase, now they're all in a line on the metaphase plate. In anaphase, they're going to start pulling apart. And then in telophase and cytokinesis, the chromosomes are going to uncondense, the nuclei are going to reform, and the cell is going to divide into two. So now we have two cells that in every way are identical to the one cell that we started with. Questions on cell division or cell cycle?
What about the organelles?
So the organelles will have to be, some of them will get divided. Some of them will have, what doesn't end up in each cell will have to be remade. Some things like the mitochondria, there's a bunch of them in there, and so when it gets cut in half, half will end up in each one, and they'll just keep dividing. Because each cell has a nucleus, it actually contains the genes needed to remake everything. So you'll have mitochondria to provide energy. Ribosomes are kind of spread out everywhere to make protein, so you can make the ER, the peroxisome, and everything else from that. Okay, so now we have to talk about genes. What's actually encoded in the DNA. And so a gene is a piece of DNA. It's not the entire strand, it's a little segment on a chromosome. A chromosome is monstrous, genes are quite small. It's a segment of the DNA that codes for a specific protein. Each gene makes a different protein. Our genome is all of the genes that make up us. We have about 20,000 genes, but that only makes up about 2% of our DNA. The rest of our DNA is non-coding. It does not code for a specific gene. Some of it plays a role in its folding around making histones. A lot of it has to do with determining which of the 20,000 genes are turned on and off. But even then, that still doesn't account for the vast majority of our DNA. At this point in time, we don't know the purpose for most of our DNA. A lot of it is probably actually more or less background. It creates an evolutionary advantage because if you have a big long string of DNA and you have a virus like HIV that comes in and it wants to insert itself, if you have a gene here and a gene here and a gene here, it's very unlikely that HIV is going to disrupt a gene. But if you have a whole bunch of genes, and then every time HIV comes in, it's going to hit a gene. So then not only are you going to have the HIV genes in your body, but it's going to ruin one of your genes. And so we think that is why there's a lot of this just background noise in our DNA. So our genes are made up of what we call the genetic code. We have the potential of making millions of proteins. So the genome is all of the genes in our body. All of the proteins in our body are called the proteome, proteome. But all of those proteins are only made up of 20 different amino acids, and they're coded for in our DNA by only 4 nucleotides. So we have 4 nucleotides that we can use to code for millions of proteins. And so the genetic code allows us to take that nucleotide code in the DNA and make it into an amino acid sequence that we can use for all of the proteins. Each amino acid has a code of 3 nucleotides, 1, 2, 3, that code for that amino acid. A specific set of 3 nucleotides, let's say ATG, is always going to code for the same amino acid. So as you read down a strand of DNA, you're going to read in triplets. 3, and then 3, and then 3. And that's going to tell you which amino acids you're coding for. And so that triplet is called a base triplet because it's made up of bases. It's either nitrogenous bases. And so that codes for 1 amino acid. The base triplet is the 3 in the DNA. What we're going to see is that DNA does not directly get used to make protein. DNA gets used to make RNA. RNA gets used to make protein. The 3 bases in RNA is called a codon. Based on the fact that we have 4 nucleotides for each position, and there are 3 possible positions, if you do the math, it comes out that there are 64 possible combinations for 3 nucleotides linked together. So there are 64 possible codons, and those 64 codons code for 20 amino acids. So there's overlap. Each amino acid is coded for by multiple different codons. There are also some codons which are what we call stop codons. There's 3 specific ones. So when we're making a protein, if you hit a stop codon, that means the protein is done. That means stop making protein. And then there's also a start codon. And it's always AUG. And AUG actually codes for the amino acid methionine. And so every single gene will start with AUG, and every single protein will start with a methionine, because AUG codes for methionine. So if I am working in a lab, and I have a strand of DNA, I know the sequence of the DNA, and I need to figure out what the amino acid sequence is, this is the type of table I would look at. And so in this table, there are all 64 codons at each of these. And if you find the 3 together that you need, let's say I had AAU, let's not use AU yet. Let's do CAC. If I had CAC, that would be histidine. And so if you know your DNA sequence, you can use that in order to get your protein. So remember that every cell in our body has the same 46 chromosomes. Everyone has the same 46 chromosomes, but the proteins that each cell makes are very different. There are some like actin and tubulin that are the same throughout, but the different proteins expressed in each cell determines why each cell is different from the next one. And so why there are different proteins in each cell is because the cells can turn genes on and off. So a liver cell is going to turn on genes needed in a liver cell, but turn off the genes needed in a brain cell. So it's only going to make the proteins it needs. And each cell usually only uses about 1/3 to 2/3 of its genes. There's no cell in your body that uses all of its genes. The rest of them just stay folded up because they're not needed, but you can't get rid of a gene. So if a gene gets turned on, and we're not going to really talk about how or why a gene gets turned on, but when a gene gets activated, it's going to be used to make RNA. And RNA, we learned, comes in a few different types. This type is messenger RNA, or mRNA. This is going to be more or less a duplicate copy of the DNA, but it's going to leave the nucleus to be made into a protein. The DNA is your master copy. You don't actually take that with you. It is saved in a safe place. If you need it someplace else, you make a copy of it, and then you take it where it needs to go. The copy is our messenger RNA. And so the mRNA is complementary to our gene. So before we said ATG would be complementary to TAC. RNA works the same. It's just what we'll see is that RNA, instead of a T, has a U. But we'll see that, I think, on the next slide. And so if we're going to make a protein, we take DNA, we use DNA to make RNA, mRNA, and then the mRNA is used to make protein. The process of turning DNA into mRNA is called transcription. Once you've transcribed your RNA, that happens in the nucleus. That RNA is going to leave the nucleus, go into the cytoplasm, and it's going to be used in the process of translation to turn mRNA and make protein. And that's going to happen in the cytoplasm. Here is a strand of DNA. The green part is our gene that we're going to use to make a protein. We're going to take that DNA and we're going to make a strand of messenger RNA. And then translation turns that messenger RNA into a protein. And then that protein can go on and do whatever that protein does. Remember that the protein is the functional part of a cell. DNA and RNA can't do anything. Only proteins can actually physically do something. And so transcription, that process of going from DNA to messenger RNA, is going to have a few different steps to it. When we copied our DNA, we had an enzyme called DNA polymerase. Here, we're making RNA, and so it's RNA polymerase, the enzyme ACE that makes the polymer of RNA. It's going to bind to the DNA, move down the strand, making the strand of RNA as it goes. And so this RNA polymerase is able to recognize the start of a gene. Genes and the DNA level normally start with TA, TA, TA. So it's going to look in our DNA strand for TA, TA, TA. When it finds it, it's going to bind. Once it binds, it's going to unwind that DNA helix, and it's going to read the bases as it goes. And as it goes, it's going to make the mRNA. If it sees a G, if it sees a C, it's going to put a G in the mRNA. If it sees a G, it'll make a C. If it sees a T, it'll put an A. But if it sees an A, it's going to put a U. DNA has T for thymidine. RNA has U for uracil. That's uracil like that. it works exactly like a T. But in RNA, there are no T's. And so it will pair a U with an A. If it sees a T, it's going to give an A. This T that it's seeing is on the DNA. So the RNA polymerase will never see a U, because it's reading the DNA, which has T's. But it will use a U in place of a T when it's making the RNA. So as it's going along, it's unwinding the DNA strand, making the RNA, and then it's going to rewind the DNA as it goes, as it's finished. But the RNA that it made is not done. That's what it just made is called a pre-mRNA, and it contains parts of the gene that are useful, and it contains parts of a gene that are not useful. So not only is the majority of our DNA not part of a gene, but buried in each gene is a bunch of non-functional DNA. And so imagine we have a new strand of mRNA, and then within that, there are some dark bands of RNA that do not belong in our functional gene. And so the exons are the parts of the RNA that are useful. These are exons. The dark parts here are what's called introns.